We are recording now. Yeah, many people from the RDRP Summit, so they're willing to repeat, to listen again to your talk. That's great.
Yeah.
Okay, I think we're there. So we're going to start. Hello, thank you everyone for coming and be very welcome to the 5th ECR Biomics seminar of this year. Let me introduce you to our speaker of today. Her name is Erin Harvey. She's currently a 4th-year postdoc at the University of Sydney, Australia, in the lab of Eddie Holmes. The Holmes lab studies the emergence of evolution of RNA viruses, and Eddie works on the viroms of Australian mammals and parasitic invertebrates, with a particular interest in effects of changing land use and anthropogenic activities on the viroms of endangered native species. Erin did her undergraduate studies at the University of New South Wales in Sydney, majoring in immunology and medical microbiology, and did her honors thesis at the Kingholms Cancer Center within the Genomic Informatics Lab under the supervision of Marcel Dinger. From there, she joined Eddie Holmes' lab at the University of Sydney and began her PhD studies in 2016. Erin began doing virus discovery using metatranscriptomics in her PhD studies and did virome studies on Australian ticks and fleas, koalas, Antarctic penguins, and their ticks. She also worked on a project using drones to capture whale blow, which we extracted total RNA from and identify novel viruses. Erin completed her PhD studies in 2019 and has been exploring her interest in mammalian viroms since then. She's currently spending 2 months collaborating with Sebastian Lequime's lab at the University of Groningen to learn more about endogenous viral elements. And the title of her talk today is The Utility of Phylogeny in Virus Discovery. host association, taxonomy, and disease potential. So Erin, whenever you're ready, you can please kindly share your presentation.
Yep. Okay, is that all good?
Yeah.
Cool. Yeah, so my name's Erin. I'm from the University of Sydney, but I am currently in the Netherlands. Hence, you can see that there's still daylight outside. If I was in Sydney, it would be midnight right now. So thank you to the people from Australia who have also joined today. And this talk will be quite similar to the one I gave at the RDRP Summit, but I will go more in depth on things like methods and some more examples of where phylogenetic trees are really useful in virus discovery. So for most of us, we are doing our virus discovery entirely bioinformatically these days, which means that we're not really left with a lot of options for analysis techniques to put our virus sequences into context. And we are pretty much restricted to comparative analysis. And in the words of the boss man, phylogeny is the obvious method. Guillermo pretty much introduced me pretty well. So my background is I've always wanted to study virus evolution, and that's why I've been with Eddie Holmes for so long in Sydney. For those who are not familiar with this figure, I think that this figure from Mengxi's 2016 paper, Redefining the Invertebrate Virosphere, really captures the impact of metatranscriptomic virus discovery on our understanding of virus diversity. And for those not familiar, the red lines indicate the viruses that were identified in this study, and the grey lines are the viruses that were in the reference database at the time. So as you can see, like, groups like the Bacorniviralis were dramatically affected by this one study, let alone all the other studies that have come along behind it. And groups like that Mono2 group that at the time was very new, now the taxonomy has completely changed entirely because of metatranscriptomic virus discovery. But on the flip side of that coin, the databases that we rely upon are still very much manually curated. When they were set up, they were sort of set up to handle maybe a few hundred entries per year, and many of those would have been resequencing of known viruses, whereas now they're receiving thousands of entries per year of completely novel viruses. And considering that they still rely on manual curation, that leads to a lot of incomplete and incorrect entries in these databases. And when we have incomplete and incorrect entries, these compound when people are relying upon BLAST alone to assign things like taxonomy and potential host associations. And this is one such example. I hope Sabrina doesn't mind me using this example again, but I came across this virus while I was looking for an outgroup for a tree that I'll talk about in more detail later. And this virus was assigned as a hand-to-like virus by the author, but NCBI has assigned the taxonomy as being within the mononegavirales. And for those who don't know, hantaviruses are actually within the bunyavirales, which is a different group of negative stranded RNA viruses. And given how divergent this virus is, I mean, it's hard to tell if it really should be within this group or not. And I have also put it in a tree with other bunyaviruses, and it does look equally as divergent in this tree as well. So when you're working with highly divergent viruses, it can be quite difficult to assign taxonomy, and just using BLAST hits can be problematic. So I'm going to go into some examples of where there are errors in the databases and how to deal with those things, and also how to use phylogenetics to analyze your novel virus sequences. So first I'll talk about an SRA mining project that I've been working on, and secondly I'll talk about a project that I did in collaboration with a group of ecologists that were sampling fecal samples from small mammals in Tasmania to look at the effects of land use on the viromes of these animals. I'll go through the workflow now just to get it out of the way because it is pretty similar for both projects. So our lab does total RNA extractions, and the kit we use really depends on the type of sample that we're working with, because our lab works with a huge range of sample types. But generally, we will go with the QIAGEN RNeasy Plus kit. That kit is pretty good. It can handle most things. And I use those for the fecal samples I'll talk about later. But I also use the QIAshredder columns with these samples as well, just to try and get absolutely everything I could out of the sample, because we weren't left with a lot to work with. We then do a ribosomal RNA depletion. And by we, I mean we send our samples off to a company called AGRF in Melbourne, and they do our library preps for us. And then we sequence on a NovaSeq. And once we get our sequencing back, we do sort of typical QC, trim our reads, and do a de novo assembly. And at the moment, I'm using a MegaHit, but there's quite a few different de novo assemblers out there, and I think there's a couple of papers comparing them. I'll go into my virus discovery pipeline now, but I just want to point out that even within our group alone, people use different methods depending on what kind of viruses you're looking for. And for me, working on mainly mammals, this works for me. So I use Diamond Blast X with a custom database of virus proteins that was put together by Justine Charon when she was in our lab. And that collects anything that's got any kind of amino acid sequence similarity to a virus protein. But this data set will be filled with a lot of false positives. So then I remove false positives with a BLAST-N against the NCBI-NT database, and I'll remove everything that has sequence similarity to something that's not a virus. So I'm left with things that have no nucleotide sequence similarity to anything, and things that have sequence similarity to virus sequences. And then with that data set, I will then do another Diamond Blast X against NCBI's NR database, and I will then take out all of the hits that still are hitting to virus proteins. But at that stage, I will look at things like the e-value and the length of my contig and the length of the sequence similarity as well. And then I will manually have a look at those contigs in Genius, and this gives me the opportunity to check for complete ORFs and to check that I'm seeing the genome structure that I would expect. And also if you're getting fragments, multiple fragments of a virus that don't assemble into a nice full genome, you can try and do a reassembly in Genius or find other missing contigs, and also with segmented viruses, try to match up and get all of the segments that you need. I will then do a web blast with those contigs, just as another check to make sure that I'm not seeing anything like host proteins or things associated with bacteria or plants or any other thing, any other false positives that could be in my sample. This will also give me an idea of what the taxonomy could be of this virus, because sometimes just using the top 5 blast hits doesn't give you a lot of information, and I'll talk about that a little bit later. And then once I've decided what taxonomy I think the virus will be, I use NCBI virus to collect all of the RDRP sequences, but that can be quite problematic, as anyone who uses NCBI virus would know, because the RDRP can be labeled as many, many different things, and you even have to account for the fact that there could be spelling mistakes. So once I have an input file that I'm happy with, I will then produce my alignment, and generally MAFT is the tool that I'll use, and you can play around with the parameters that you use, but sometimes if you're working with particularly more divergent viruses, you might need to play around with the tools that you use and explore something like Clustal or even the aligner in Genius. I'll then trim my alignment, and I always manually assess my alignments to see if the alignment tool I've used looks like it's the correct one, if it looks like I've taxonomically assigned my virus correctly, and also to see if I need to change my parameters. And then once I'm happy with my alignment, I will put it into IQ-Tree and use Model Finder, but I will always check what model it has picked for me and make sure that it makes sense in terms of what I would expect it to choose. And I got a question at RDRP about how you decide if the model is right, and all I can say is Google is your friend, and to read other papers where people have built trees on the same sort of family. So now we're getting to the interesting stuff. So the first project I'll talk about is this SRA mining project, where we screened all of the dasyurid RNA-seq libraries that were on the NCBI SRA, and that was 446 libraries. And the dasyurids are a group of Australian marsupial carnivores, and marsupials are mammals that have like a pouch that they grow their young in, and they're very common in Australia. And so we passed this through a more liberal pipeline that is put together by my student, John Mifsud, and then I put it through my pipeline, which is a little bit stricter just for personal preference, and we removed all of the eaves. And we know quite a lot about the eaves in marsupials thanks to Emma Harding's paper. So we identified 15 novel viruses through this screening, and surprisingly, most of them are actually DNA viruses. And I think this could be because of these libraries were generated not for the purpose of virus discovery. And so there could have been steps in the library prep like polyase selection that would remove a lot of viruses. And so, but we did identify 2 Delta viruses and 3 RNA viruses, and these were across numbats, antechinus, fat-tailed dunnart, and Tassie devils. And you can see there's an overrepresentation of the antechinus and the devils, and that's indicative of how many SRA libraries there are available for those species, and they are more studied because of their biology. So I'll start off with talking about these Delta viruses. Up until quite recently, Delta viruses were thought to only infect humans and to have emerged in humans. And that was proven to be incorrect through metatranscriptomics. There was a Delta virus identified in birds by my colleague Michelle Wiley, and one in a snake, I think. And since then, SRA mining, particularly using the Serratus web tool, has identified a few other mammalian Delta viruses. And we identified these 2, and they form a little monophyletic clade, which is quite interesting. And I just want to point out that I did use Laura Bergner's paper as a reference to build this tree. So even if you do have a lot of experience in phylogenetics, sometimes it is best to look at what somebody else has done to help you, to help influence the way you build a tree, because building this tree can be quite difficult. But yeah, so it'll be interesting to see if we can sequence more marsupials and see if this sort of monophyletic clade holds up. So we also identified 2 novel hepaciviruses, one in an antechinus and one in a numbat. And the antechinus virus clusters with a bandicoot hepacivirus that I actually identified from a blood meal of a tick a few years ago in my PhD. And of note, the marsupial hepaces don't cluster together. So there's also this cluster down here of a koala and a possum. And my theory is that this difference could be because bandicoots are omnivores, and they also sort of scrounge around on the bush floor, and so do antechinus, whereas koalas and possums are tree dwelling and they're herbivores. So this difference in hepaciviruses could be that there's been multiple spillovers into marsupials, and that could be due to their differences in environmental niche. But you might have noticed the other novel virus is sitting all the way down here, and that's our numbat herpessyvirus. And numbats have quite interesting biology in that they exclusively eat termites, and they are the closest relative to the Tasmanian tiger. And they are now only exist in 2 national parks in the southwest of Australia. But that doesn't really explain why this virus would be clustering down at the bottom of the tree with a group of viruses that is associated with birds and reptiles. And I think that could be just that this virus is so divergent that there's not a lot of bootstrapping support for these nodes down here. And so until we sort of fill in that evolutionary space between it and the other mammalian viruses, we don't really know where it actually sits in the tree. And as you can see, the herpessyviruses do generally sort of cluster by host. But you may have noticed this blue line down here that sort of bucks that trend. So I noticed this virus while building this tree, and this virus is listed in NCBI as having a mosquito as its host. And for those who know anything about herpessyviruses, you'll know that they're not vector-borne and they're not associated with invertebrates. So this virus looks quite suspicious. And given the fact that it has come from sequencing of a mosquito, I would be concerned that this may have actually come from the blood meal of that mosquito rather than replicating within that mosquito itself. And I think that's where we need to be careful when we assign a host to a virus, because should someone come along with another novel virus, they might assume that it could be vector-borne or invertebrate infecting, when in fact this is just a mistake. But on the flip side of that, sometimes you also identify highly divergent viruses that buck that trend. And this virus that was identified in a library of devil facial tumor disease, which is a cancer that affects Tasmanian devils. I 100% thought this was contamination to begin with. And this is where the benefit of our whole RNA sequencing approach comes in, because we can actually have a look at the composition of the whole library to see if there's any potential sources of contamination that could explain this virus. And so this was cell line sequencing, so it's kind of unlikely that we would have any sort of invertebrate here, but you never know, maybe a fly in the lab fell into the culture, or I don't know, anything could happen. But we didn't see any source of contamination. We use a program called CC-Metagen to have a look at the composition of the libraries, and it spits out a nice little chronograph. So you can sort of have a look around and see if there's anything that could be potential contamination. This virus was also quite abundant. It was the most abundant virus in this study. And this might not seem like a huge abundance, but for viruses, this is pretty impressive. And it's also extremely divergent, with only 24.3% amino acid sequence similarity at the RDRP. And that was to an invertebrate virus. It's also looking like this virus is exogenous. We have a nice long contig with a really nice ORF. But 2 viruses were discovered in invertebrates, and thus far they have largely been only identified in invertebrates. But recently there has been the description of a new genus called the Piscichu, which includes some fish-associated viruses and 2 viruses from snakes. There's also a paper that I'm not sure if it has been published yet, but is out as a preprint, where they identified chew-like viruses in turtles. And a lot of these have not only a disease association, but an association with neurological disease. And there are eaves of chew-like nucleocapsid proteins in Tasmanian devils, but that doesn't look like it's anything to do with our sequence. It's quite divergent from our sequence, and it's full of stop codons, whereas we have a nice big ORF. And it wasn't, I was still on the fence about this and thinking that it was most likely contamination of some sort, until my PhD student, John Mifsud, reviewed the manuscript, and he came across a paper that suggested that DFTD is of Schwann cell origin, and Schwann cells are part of the PNS. So this holds up this idea that maybe vertebrate chew viruses are associated with the nervous system, and definitely makes me feel a little bit better about suggesting that this could be the first mammalian chew virus. So now on to the other project. So our ecologist collaborators from Tasmania came to us with the question of whether things like agricultural, land use, forestry could have an impact on the viromes of native species. And so they went out and did a whole lot of trapping. So these fecal samples were all taken from trapped caged animals. So we can be certain that the fecal sample is actually coming from our target host. And they looked at feral animals like cats and black rats, as well as native species, and that included carnivores like devils and quolls, as well as herbivores like brush-tailed possums and swamp rats. And so we extracted all of the RNA from these individually and then pulled them by collection location, land use type, and species. And you might notice on our map down here that we've sort of excluded this southwestern area of Tasmania. And that's because that's one giant national park, essentially. And it's actually where Alone Australia is filmed. So if you want to see what the environment actually looks like and what some of the wildlife look like, I suggest checking that out. So we identified a whole lot of virus contigs in our libraries. Because we're looking at fecal samples, that's sort of expected, particularly when you're looking at carnivores, because they're really scavengers, so they get into everything. So I decided that maybe it would be more useful just to look at mammalian associated viruses in this case. And I'll talk a little bit about how I decided what to include and what to exclude. So for the sake of time, I'm just going to talk about the Picornavirales, because we identified quite a few interesting species in here, but also because I think it's an order that people have quite a lot of problems with understanding the taxonomy, and there's quite a few incorrectly identified Picornaviridae that are actually just Picornavirales. So it's important to note that there are only two families within the Picornavirales that are mammalian associated, and that is the Picornaviridae and the Caliciviridae. So it can be quite difficult to determine if your novel virus belongs within one of these two families. This is the top blast hits of one of my novel viruses. And as you can see, the top hits don't really tell us a lot. The top hit is my pet hate, Riboviria sp. But we also have some hits that look like they could be real Picornaviridae, hepatovirus, gallivirus, enterovirus. These are all Picornaviridae. But we also have a real, weird result here, seeing some Nanaviridae sp, which are a completely different family of viruses that infect fungi. So I put this blast result in a tree, and as you can see, we have our Nanas clustering up here at the top, the Picornaviridae clustering here, and then our more divergent, more invertebrate associated viruses down the bottom here. So it does look like, despite the second hit being a hepatovirus, our virus is probably not vertebrate associated. And so this is one that I would have excluded. And just on a larger scale, this is how I identified. So first I would use a blast screen to exclude the things that are obviously not Picornaviridae. But then with those things that had sort of confusing top hits, I would put them in a tree and use that to exclude things. This tree is obviously very, very difficult to look at from this resolution. But you can see there's a few sort of red lines around there that are in the wrong spot, so they would be excluded. And again, for the sake of time, I'm just going to talk about the Caliciviridae that we identified in this study. So we identified 7 caliciviruses in our study, 2 of them having been previously identified, and that is canine BC virus and RHDV2. And this was in a range of samples pretty much covering all of the species that we sampled here. And this includes feral and native species, as well as herbivores and carnivores. And so because a lot of caliciviruses can be associated with gastrointestinal issues, we asked our collaborators if they saw any signs of disease, because they had actually tracked the animal. They had done a sort of general health check, and also because they're sampling feces, they could tell if there were any signs that something was wrong. And they said there were no signs in any of the animals that they were sick. And so it's unlikely that things like this canine VC virus are actually causing disease in Tassie devils. Sorry, TD is Tassie devil. But it's very likely that this is rather coming from a terrible habit that Tassie devils have, and that is eating the feces of other animals. And we also identified a lot of human-associated viruses, like human rotavirus, in the Tassie devil feces, and we think that's because they go to campsites and get into the sewage systems, which is pretty disgusting. So don't be kissing Tassie devils. And across the other species, it's quite difficult for us to say whether these viruses are truly infecting our sampled host or if they're coming from the diet. But in the case of the brush-tailed possum, considering that they're a herbivore, we might be more likely to suggest that this could be coming from the brushy. But you never know, because they'll also get into anything they can get their hands on. The most concerning thing that we found here was RHDV2, and that is rabbit hemorrhagic disease virus 2, which is used as a biocontrol agent in Australia to control the rabbit population. And there has been quite a lot of concern that this virus could jump into native species, and if it had the same effect on native species as it does on rabbits, this would be quite concerning, because a lot of native species are already at risk of extinction, and a virus like this could completely wipe them out. So that's when it's important to look at the biology of your virus. And RHDV2, it affects the liver of infected animals, and you would not expect to see this in feces. But we're lucky enough to have an expert on RHDV2 in our lab, Jackie Ma, and she built this beautiful tree for me, and showed that our RHDV2 was indeed clustering with other RHDV2 isolates taken from rabbits in Tasmania. And it's not looking like this virus has changed significantly in order to jump into a new host. And the fact that it's only been found in feces also suggests that it's not infecting the host that we've sampled, but rather is coming from the fact that the animals that we've found this in are scavengers. And so they're finding dead rabbits and eating them, and they have a high titer of virus. And this was concerning because we actually had 25% of total reads being RHDV2 in one of our devil libraries, which is an absolutely insane amount. So I'm just going to summarize what we're doing and what we're doing in the future. So the marsupial carnivore SRA paper should hopefully be out soon once Eddie gives it the okay. And the second project I've talked about, we're actually doing ecological analysis and using VOTUs for that, and that's for the purposes of our collaborators. But then I will also do a virus characterization paper where I put all of these viruses into trees and follow the ICTV rules and make all of those sequences available online, and try and provide some context. We also have blood samples collected during the fecal sampling, which will be quite interesting if we see any overlap between the viruses we see in the feces and the viruses we see in the blood. But it's not a perfect overlap, and we do have some other species as well, like bandicoots, which will be interesting to look at. And I also want to use the Serratus palm scan tool to try and see if I can identify any more chew-like viruses similar to my devil chew virus in any other mammalian libraries. And just since I have a little bit of time, I just want to advertise my PhD students and the lovely work that they've done. So this work was done by John Mifsud. He's the guy on the left. And he builds these beautiful tanglegrams to look for cross-species transmission events in the viruses he identifies. And so he extended the host range of the Flaviviridae using SRA mining. So he found pestiviruses in amphibians, reptiles, and ray fin fish. And he also identified a potentially novel cross-species transmission event between bats and rodents. But he wants me to say that it's very potential. And also Vince, who is Mr. Fish in our lab, he put out this beautiful paper looking at doing sequencing of fish collected from an area of 100 square meters in the Great Barrier Reef. And he was looking for signs of cross-species transmission in the viruses that he identified. And he saw little evidence of cross-species transmission and saw that there was more diversity in the smaller fish. I'm not going to try to name them, but which is interesting because they have very short lifespans and grow very rapidly. And so perhaps there's a bit of a payoff between that, between the rapid growth and being able to fight off viruses. And he also identified a couple of viruses in this PSQ2 group, and he'll have a paper coming out hopefully soon that he's done in collaboration with a group in Switzerland where they've sequenced a whole lot of cichlid fish from a single African lake, and he's looked at the viruses in those fish and looked for patterns of cross-species transmission in those as well. So just to summarize, we're pretty limited in the type of analysis that we can do when we're just working with virus sequences. And I think phylogeny is a great way to get a lot of information with quite minimal input, and also using tools that are very well described and easy to access as well. I want everyone to keep in mind that you can't just trust everything that's on NCBI, and that building trees is a great way to check things like host association and taxonomy. And if you're very new to phylogenetics, don't be afraid, read lots of papers. A lot of people are now publishing their alignments when they publish their papers. So I would suggest looking at those alignments, trying to recreate them yourself, but don't be afraid to ask for help. And there's a lot of courses out there as well to keep an eye out for. So I just want to thank Mena and Antje, who provided me with the fecal samples, and everyone in the Holmes lab.
Thank you so much, Erin. Incredibly interesting talk. So the chat went busy really soon, so I'm just going to start scrolling up to see who was first asking. Yeah, Jean-Claude Macangara, you have several questions. Would you mind to go over them?
Yep, that's fine. So the first is for...
Oh, I mean, I was asking the person who
sorry.
was the question, or if he's not already there. Jean-Claude, are you there?
I think yes.
Thank you.
Yeah, thank you very much for the presentation and for this topic, which is really interesting. Hopefully, I'm working also in the on the same topic, but I'm working on protocol of metagenomic to discover novel viruses, and I'm using nanopore. But also, I'm planning also to develop a bioinformatic pipeline which can be easily deployed in a limited resource region. So that's why I have many questions about how you proceed. Maybe the first question was about your protocol, your bioinformatic protocol. How do you proceed? How do you have you customized one pipeline, or you just go from one pipeline or from one tool to another tool. And after that, when you have your outputs, then you can go for phylogenetic analysis.
Yep, so we're lucky enough to have access to the University of Sydney server. So we have quite a lot of computational power at our hands, and also a lot of storage space, which helps with a lot of the SRA mining projects, because obviously if you're downloading all of these libraries and assembling them. But I do think the most efficient way is when you're downloading, after you go to the next step, have a well thought out pipeline that deletes the file from the previous step when you no longer need it. And that way you cut down on your storage requirements. We have automated pipelines that go from the step of trimming, assembly, all the way through to like a final data set that'll be in the form of something like a CSV file that we'll then manually have a look through. Unfortunately, because depending on what types of viruses you're looking at, a lot of the reference databases are becoming more and more like muddy, and it's harder to automatically pull things out based on sequence similarity. Like that CHU virus, there's no way I would have pulled that out if I had an automated pipeline, because I would have thought that CHU viruses were invertebrate associated. But also you could be getting a lot of false positives if you're using the reference database of, say, Picornaviridae, because there's a lot of non-mammalian sequences in there. So unfortunately, I don't think it's possible to completely have, you know, one lovely pipeline that just spits out a tree at the end. But that first section of our workflow is in a pipeline. And then for the phylogenetics, again, I think it's at the moment quite manual. It would be great to try and develop some sort of method, maybe through utilizing something like AI, to try and build your input data sets, because it's quite, unless you're always looking at the same families, it's quite difficult to build those input data sets because so many viruses are unclassified. So potentially the viruses most similar to your virus might be missed if you're just automatically pulling down by assigned taxonomy. Does that answer your question?
Yes, I think, thank you. Another question was about the method you use for RNA depletion, rRNA depletion. And maybe I could go also, do you only look for RNA viruses or also DNA? Another question. So maybe I can finish and let other people going on. Do you do the protocol? Your protocol, is it for the whole genome, or you just look for reads which are related to viruses and then you store by there? Or after having those reads, do you also develop or maybe primers or to look for to amplify the whole genome? Over.
Yep. So for the rRNA depletion, that's done using the Illumina kit, but we don't do any of that in-house because it's quite difficult to get a good setup for building libraries. We find that it's easier to outsource that for us. Also, so we sequence everything. We just do a ribosomal RNA depletion, and then we do short read sequencing. So we're not getting full genomes. We have to do that de novo assembly step. And you're not always going to get nice full genomes. Sometimes you will just get fragments. Depending on how interested you are in the virus you found, we might go back, design primers and PCR. And the viruses in Vince's paper, those have all been PCR'd, I think, except for one where he didn't have any sample left. But yeah, we do incidentally identify DNA viruses just because you see those transcribed genes sometimes. And I actually found more DNA viruses than RNA viruses in that SRA screening project, which is interesting. but you never get the full genome, obviously. So then sometimes it's better to go back and do some targeted DNA sequencing if you want to try and get the full genome. But obviously, when you're working with SRA, you have no options.
Thank you very much. Maybe for other question, I can send you an email.
Yeah, perfect.
Thank you, Jean-Claude. So next question was formulated by Luis Tataje Lavanda. Would you mind to read your question, Luis?
Hello.
Hello.
Hello. I'm from Peru. Sorry for my English. I prefer to read my question. Thanks, Erin. It's very impressive your presentation, very informative. Especially in my case, I am very interested in this kind of study as metagenomics. I try to start to study for the development or to find new variants of virus. Virus not only for men or people, too for animals. In this moment, we have nanopore technologies that quality is improved, no? before not so good. But my question is more about how do you determine that the sequence is from a specific new virus, or maybe could be an artifact, because I think the different programs or software have determined parameters and could be generate errors into the de novo assemblies. How is your perspective for determining could be real or not real? What is the cutoff?
Yep. So again, it's quite a manual process of looking at the config that we get. Are we seeing a full genome? What sort of fragment? How big are the fragments we're getting? I tend to eliminate anything that's too short. So anything shorter than say 600 nucleotides is definitely out of the question. We also always look for a complete RDRP if possible. You also, I know this is the most annoying thing to say, but you get a feel for it once you've been doing it for a while. And that's unfortunately just through following false leads. And I think once you put things into trees, you get a better idea as well. But generally, I think to begin with, you want to be getting nice full genomes, and you want to be using your BLAST pipeline to look for sequence similarity to other viruses. Also, depending on your host, there might be information out there about endogenous viruses. And I'm actually over in the Netherlands to work with Sebastian Lekeem on trying to find out a way that we could potentially do some sort of scoring system to see if a virus that we find through metatranscriptomics is likely exogenous or endogenous. But it is very difficult. Unfortunately, it does come down to a bit of a manual approach. Does that answer your question?
Yes, thanks. And other question is number 4 is: do you think nowadays we can find a new variant of virus on databases SRA and ANA? Is possible, and is possible to found to put in a new paper? Is valid?
Yeah, I think so. Our lab has done a couple of these types of papers, and Artem's tool, Seratus, is very useful as well. So that's a web-based tool. So if you have a particular virus family that you're interested, you can use Seratus to screen the SRA for particular RDRP signals, and then you can reduce the number of SRA libraries that you have to screen. So instead of looking at absolutely everything and assembling and analyzing thousands and thousands of libraries, you can sort of target your approach more to just the libraries that have some sort of signal. And I think it's very valid to reuse somebody else's data. Most of the data that I used in my mining project has been developed for looking at the facial tumor in devils, and looking at the biology of antikinases. And then the NumBat one was because they were doing a genome and they wanted a transcriptome to match it. And there were only 3 libraries and we still found that herpessyvirus. So I think in the same turn, like when we do any sequencing, we put our libraries up so that if somebody else is interested in maybe the plant viruses or the invertebrate viruses or something to do with the transcriptome of that animal, they can use our data as well. So I think, yeah, I think it's very valid to be able to use SRA. And even if you're doing a sequencing project yourself, I think it's a useful tool to go and look at what's also available, because you might find other things there as well.
Okay, thanks. Very informative.
Thank you. Next question is from Ella Tali.
Hi.
Hi.
Nice to see you again. Yeah, I wanted to ask about your reciprocal blast approach. So
yeah,
I work in soil, and diversity of viruses in soil is just ginormous. And running Diamond, I mean, Diamond is fast, but it's just not very sustainable. So I was wondering, what's your opinion about doing the same thing but with HMMs instead? So you could get HMMs of viruses from GOG or any of the RDRP tools, and then to eliminate false positives, you can use PFAM or KEG. So what's your take on this?
Yeah, I definitely think that that's probably the way forward for big studies like that, especially when you're looking at those really, really, really divergent viruses that have never really been studied before. I think Justine Sharon and Sabrina Sadiq are probably better people to talk to about that. They're also from my lab, but yeah, within the sphere of looking at mammals, you're really, looking at a very narrow window compared to the true diversity of viruses.
Thank you. It was a great talk.
Thanks.
Thank you. I think this question is already answered from Jean-Claude. Luis Jansen has a question, I think.
Hello, hi, Dr. Harvey, great talk. Please forgive me if I didn't understand some part of this, but you found some viruses associated with Tasmanian devils, and you've mentioned at a brief point the transmissible cancers. And so I work with the viral discovery tool with Viromics, but also I have a little foot on viral epidemiology. And so I was wondering whether the 2 viruses or any other virus you found could have any sort of epidemiological link with the cancers, given that these cancers are a major threat to the conservation of these species. If there is any sort of plans on trying to elaborate, like if any of these viruses can be used as a marker for the contacts with diseased animals and so on.
Yeah, so there were quite a few libraries in that study where we identified the 2 virus, but it was only in one library. Off the top of my head, I couldn't tell you how many were sequenced in that one project. I also don't really know the structure of their project, but I would definitely, the reason that I started this SRA screen was actually to look at devils and to see if there were any potential viruses that could be, contributing factors to the pathology that they see. And I think that 2 virus, like considering that it's associated with, neurological disease, and then we've got it in this cancer that's associated with PNS cells, maybe I would love to do more work on it. And if anybody listening has some ideas or some way of getting samples and doing some screening. We also identified papillomaviruses, a polyomavirus and a herpes virus in devil samples as well, but they were from lip tissue, and they were from both individuals that had DFTD and individuals that did not have the cancer, and there didn't seem to be any kind of association with or without the disease, but obviously that's very, very scraping the surface there, and it would be very interesting to do more work and maybe design some primers so people could check their samples.
Okay, thank you.
Thank you. So next question is from Alejandro Olmedo Velarde.
Hello, Erin. Nice presentation, Jay. My question, it was two questions. So the main thing is you are using MegaHit, which was designed to deal with metagenomes, not necessarily metatranscriptomes.
Mhm.
So I wonder if you have compared that with a tool that was designed to deal with metatranscriptomes, such as RNA spades. And second question goes for the phylogeny side. You said you use trim-al, and I never heard about it, so I would like to try it. Normally, I have to do like trimming the alignments manually because some tools like are too strict with some virus proteins because they are just too divergent. And although, let's say 200 amino acids, you end up having like only 20, 30, something like that. So those are my two questions.
So the people in our lab have all sort of compared different aligners. I used to be using Trinity, which is obviously just like way too much memory requirement and not at all designed for a metatranscriptome. I've had good results using MegaHit. I haven't personally used RNA spades, but I think other people in our lab have. Maybe I should compare those as well and see if I get better results. And in terms of Trimble, it also depends on your alignment. I would say an alignment of only 200 amino acids is already very short. I normally like things to be over 300 and like really, really would love it to be over 600 amino acids if possible. When you're working with things that are really divergent, I don't really know what to say because I myself have struggled with this problem as well, trying to produce a nice tree. But I think you need to play around with your alignment algorithm as well. Maybe try some different aligners like Clustal. I think Mary Petrone from our lab was saying that it handled some of her divergent viruses a little bit better. And yeah, I think Trimble's also going to, you can set parameters and tell it how much you want it to get rid of. So you can say, I want it to keep, 60% of my alignment or 5% of my alignment. And you can also set whether you want a gappy or no gappy output. So I think playing around with parameters is the first port of call, but maybe also check that you have all the unclassified viruses in your tree as well, because they might be more closely related to your novel sequence than things that are in ICTV or classified in NCBI.
That's great. Thank you so much.
Thank you. Frank Onyambu, do you want to read your comment, or it was just a comment more than a question?
Mine was more of a comment than a question. I was very impressed by the first project, that a project will come out of mining RNA-seq data and actually discover viruses from that. Seems like something that we should be able to replicate in places where we don't have facilities to do those kind of studies. So thank you. Very good presentation.
Yeah, for sure. I think it's a great resource to be able to mine the SRA. And I promise I'm not biased because I'm friends with Artem, but Ceratus is also a really great tool as well.
Okay, thank you. So the next question is from Kane.
Hello, can you hear me?
Yes.
Yep.
Okay, thank you. I have a couple of questions. The first one is recently during the previous work, I have it was a bad sample. And, there is a group of viruses, maybe not really known on the field of virology, but recently it's gaining more and more interest. It's CREST DNA. So there is, I can say one or two, especially the circovirus group that is well known because the recurrent epidemic in pig farms, but there is a lot of unclassified viruses there. So recently when I was working, I get some sequences from megahits assembly. And when I did BLAST, I used Diamond BLAST, and I go back again to do the BLAST manually. And I found some of these sequences have been labeled with, for example, one of these sequences that really tricked me is the sequence was clearly sickle viruses from NCBIE, and the percentage identity was around 95 to 100. So, but when I construct the phylogenetic, and I found that it's totally another, it's clustered to another family, but not circoviruses. So how this kind of issue happens and how they might impact in virus classification, because it's okay for highly similar sequences you found, but what about those sequences? I still have many of them, but still, even when I take the whole order of Chris DNA Vekota, and I went to ICTV metadata, and I download all representative species there, and I made the whole phylogenetic tree based on the rep protein, and I see most of these sequences that was mislabeled was completely unclassified viruses. So how to solve this issue that are still pending there, and that might create more and more difficulty for phylogenetic relationship? And
yeah.
Can I finish, or just wait
Yeah.
Really briefly, because we have only 2 minutes.
Okay. So the second question is how the length of new virus sequence impact the reliability of phylogenetic relationship, because sometimes we use short sequence, sometimes length sequence, sometimes partial RDRP. So how this issue can be addressed? Thank you so much.
Yeah, that group of viruses, the circos, are quite often contaminants as well. So we do no template controls where we use the we'll do an extraction that has no input essentially with the kit, and we sequence that on its own as its own library, and we identify circoviruses in there. Ashley Porter wrote a paper about it. And so circoviruses, I think, can quite often be contaminants. So, they might be also misassociated with a host when really it's coming from your sequencing library. And I think that whole group is a complete mess, to be honest. I have the ability to avoid them, thankfully. I really don't have the answer for that. And at RDRP, we talked about these kinds of issues at length, the fact that so many viruses are misclassified, and how do we rectify that? And I think you can always write to NCBI, so you can email them and say, like, this virus is actually misclassified, and then they will fix that. But I'm sorry, I don't have the answer for that. I think that's a really complex question, and it's only going to get worse as more and more people are doing metatranscriptomic virus discovery. And your second question, you definitely want as long of a sequence as possible. As I said, lower than 300 amino acids, I think, is not a very robust tree, and you'll see that in the tree that you get. I think, yeah, I don't know that there's really an answer to that question in terms of how to deal with it, other than to maybe resequence PCR, try and get more of a genome, and at least try to get a lot of that RDRP segment. But yeah, I don't know that there's necessarily a way of doing phylogeny with anything shorter than that. Maybe if you're getting that key signal of the RDRP, which that could be useful. But yeah, I'm not really sure.
Okay, one quick question is what is the real benefits for metatranscriptomic compared to metagenomic for new viral discovery? I think that's really interesting.
It's a lot easier to get rid of all of the host-associated stuff. So you're getting rid of all the host genome, the host DNA, and you're just looking at the RNA. And when you do a ribosomal RNA depletion, you cut down the amount of non-virus RNA that's in your sample dramatically. I mean, you can also do a lot of other methods like doing filtration or yeah, there's loads of different methods, but we just our lab does total RNA sequencing, and we find a lot of viruses. Whereas DNA, I haven't done it myself. I think you would have a hard, maybe if you know the host. So say you've got a well-described host, you might be able to remove the host genome from your sample. But most of the things I work with don't have a genome available.
Thank you so much.
Thank you, Ken. Thank you, Erin, Harvey. So interesting. I think we could be here the whole evening, but we need to finish now. Thank you again. I will see you very soon in the next seminar. Thank you.
Thank
Bye-bye.
you.
Great job, Erin. That was a really great talk.